Hi. In this first video, I thought I'd offer a personal perspective about the place of biology and biomedical engineering in modern society. I'll start by giving a historical perspective. There have been certain times and places when human history reaches a turning point and the trajectory of civilizations are altered. I believe we live in such a time right now with respect to the biomedical sciences. With the deciphering of the human genome, the emergence of stem cell technologies, and the tools of genetic and genomic engineering, we have the potential to significantly increase health, longevity, and human capabilities while decreasing disease, and indeed will even have the ability to transform what it means to be human. Will these developments turn out to be a good thing for humanity? History will be the judge of that. Speaking of history, and since we're at a liberal arts university, do you know what the revolutions or epochal events that the dates and places here refer to? Spend a few minutes looking those up, and we'll spend a few minutes in class checking your knowledge. Now, in contrast to these previous events, which did have a fair degree of geographic localization, Athens, Rome, Florence, the current movement transforming biology is truly international. And I only put Boston there because of the concentration of universities, research headquarters of big pharma, and all the biotechnology firms in this area. It's really true that for any existing biomedical problem, it's likely that one of the top three of the world's experts in that area is just a cab ride away in Boston. No other areas have that kind of concentration of skill and expertise, and that's another thing you should be taking advantage of while you're here. Many advances in human society, or great leaps forward for civilizations, are the result of technological advances. I've drawn up my highly unofficial list of seminal inventions and discoveries that I thought were the most important for shaping society as it is today. I invite you to look at a recent compilation of the top 50 inventions compiled by Atlantic Magazine. We might discuss a few of these briefly in class. Spend a few minutes thinking about two of the biggies, fire and the printing press. Do you know why fire was such an important technological advance for the progress of the human species? I'll give you a hint. It's not for warmth in colder climates. It's something much more fundamental. Bring your best ideas about fire to class. I'd like to hear them. I'd also like to hear what you think are the important inventions and discoveries that I've left off. Make your case for the important things that I've left out, and we'll discuss them in class. The point of this slide is to motivate the next one, which is once again my highly unofficial list of the top 10 biological developments of the last half century that has helped to establish modern biology as it is today in 2014 and will shape it going forward. These are in chronological order. The discovery of the structure of DNA, the double helix, reveals a tremendous amount of biology. It made clear the way information was stored in the hereditary material. It suggested how DNA was replicated to make copies of itself, and it led in short order to the elucidation of steps needed to make RNA and then protein. The next major development I have listed is recombinant DNA engineering. This is the ability to take a piece of DNA, digest it, cut it with a restriction enzyme so that you can have a molecularly defined piece of DNA. You could then clone it into a plasmid and grow it in bacteria and have millions of identical copies of that piece of DNA to study. Prior to recombinant DNA, it was impossible to work on the same piece twice. Long pieces of DNA were sheared and they would be fragmented randomly. And now with recombinant DNA, we can start to engineer and manipulate DNA in ways that will help us study the DNA and produce desired effects. DNA sequencing, which was first developed by Frederick Sanger, Walter Gilbert, and Alan Maxam, let us decode the information stored in DNA, the sequence that held the information. This sequencing technology has evolved over time and is now improving faster than Moore's law for transistors and has led to the possibilities of a $1,000 genome. Monoclonal antibodies is the first time that we were able to produce immortalized antibodies, essentially creating an infinite supply. And this was critical for the standardization of protein studies. Now, a researcher in Boston and a researcher in California could say, I'm studying the protein that reacts with this antibody and weighs about 50 kilodaltons, and it gave a great deal of certainty that you were talking about the same protein. PCR, developed by Kary Mullis in the early 80s, greatly increased our ability to manipulate DNA, basically turbocharging recombinant DNA, the ability to amplify a single DNA molecule into billions of copies that can be sequenced or cloned or used for any particular purpose. We couldn't have modern biology without the development of personal computers. There's so much information that no human being can store them. The data sets have grown so large that, like big physics, single biological experiments can now include millions of data points, and the only way to keep track of is with the use of computers. Microarrays, or DNA chips, developed in the mid-90s, was a major step forward in enabling the global or systems level study of a cell or organism, where we could keep track of each gene and think about how the parts relate to the whole. Prior to this, biology was extremely reductionistic. The goal was to study and isolate a single protein and learn all about it, and then trying afterwards to fit it back into a picture of the cell's operations. But microarrays essentially enabled the ability to take snapshots of what every part in the cell is doing at a particular time. Two events in the late 90s, the cloning of Dolly the sheep and the isolation and growth of embryonic stem cells, have hinted at the immense potential of what can be achieved using these very powerful biological techniques. We are still in the earliest days of these technologies, and no doubt they're scary to many people, but they have so much promise for the eradication of disease and the enhancement of human health that they will undoubtedly be the biggest story 100 years from now. Then in 2003, the completion of 97% of the human genome sequence finally gave us a picture of what it takes to make a human. It's been over 10 years since then, and we have miles to go before we achieve that level of understanding. But I think it's probably not too long until you have your genome sequenced, and then it'll be a little longer and we'll be able to interpret what that means. But with the greatest hope of perhaps anticipating disease or treating diseases that one might develop, or changing lifestyles to prevent certain diseases from ever occurring. Now in class, I would like you to come prepared to class having thought about some of these and develop what you think the top 3 most important developments are. Biomedical engineering itself is undergoing a tremendous change in terms of how the field is defined. It's probably safe to say that 10 or 15 years ago, the only approach to fixing a failing heart that a biomedical engineer would consider is the implantation of some sort of device or instrument to help assist the heart, like a ventricular assist device, a pump, or a pacemaker. Nowadays, it's equally appropriate to think about using stem cells to repair a heart, to regrow healthy heart tissue, to do genetic engineering, to change the behavior of certain cells in order to have improved performance. It is extremely important for you to not look at these two approaches as opposed to each other. As an engineer, your goal should be to fix a broken tissue. It doesn't matter what methods you use. It matters how well you fix it. If you take a narrow viewpoint and insist that only devices or instruments is truly biomedical engineering, you risk defining yourself into obsolescence. The best methods will prevail, and you might as well cast a wide net. I'd say the same for the delivery of a drug compound from a pharmaceutical company. Don't think of that as the realm of biology. of a person taking a drug. You can think of it just as well as that you're an engineer who is giving a inducer molecule to perturb a system from an unhealthy state to a new, healthier state. You're changing, engineering, and fixing a system by the delivery of a small molecule. That is biomedical engineering. Now, this is sort of the bummer slide. This will be a very challenging course. Biomedical engineering is basically a double major. It's not good enough to know a little bit of biology and a little bit of engineering and think you can provide something special by having a little bit of overlap between the two disciplines. You really have to aim for knowing as much biology as the biologists and as much engineering as the engineers to fully capitalize on the path that you have selected for yourself. If you do that, you will be in a fantastic position. You will have biological and quantitative skills that neither the engineer by themselves or the biologist by themselves will have, and you will have a unique viewpoint on biological problems and a unique skill set to attempt to fix those. Now, my goal is to make this course as simple as possible for you to learn a great deal of material. The lectures are organized with the book, and the tests are developed straight from the lectures so that there will be no surprises. Important things will be emphasized over and over again and will be the bulk of what you're tested upon. If during the course of this class you can think of ways that we can make things easier for you, please let me know. I am eager to do it. I can guarantee you with even a medium amount of effort you will learn a lot of biology this semester. That said, I have to confront a bias that people from physical sciences tend to have. They tend to look down on biology because a lot of memorization is involved. I urge you to not fall into this trap. It is a defeatist trap. Facts are important. I would use the analogy of a lawyer in a court case. You might be the most brilliant lawyer and most persuasive speaker, but if the other side has a fact that disagrees with your arguments, you're going to lose the case. In biology, you don't derive a lot of phenomena based on fundamental principles. It's more the collection of thousands of facts and the development of concepts that help explain those thousands of facts. With regard to memorization, in second or third grade, you memorize the multiplication table, and that has been tremendously useful for you over the course of your career so far. So too will it be if you memorize a number of the basic facts that we'll discuss that are contained on the concept list, which I'll make available, that will serve you well as in your future careers as you pursue biomedical research. On the next couple of slides, I have a few articles from the popular press that talk about different disciplines of study. On this one, we see why literary criticism matters. Here's an article asking, why should we teach English? And then here is a debate on the fate of the humanities. The reason I bring that up is because no one ever asks these questions about science or engineering. Can you even imagine an article in the newspaper that would say why it is important to study science? It's so fundamental that it's not even questioned anymore. Now, that's not a reason to gloat. Just like biology and engineering are not in competition, the sciences are not in competition with the humanities. The reason I mention it is because it attests to the fact that modern society knows that science and engineering matters. It is a given. That has certain consequences. Because it so matters so much, no matter who you are, if you have insights, if you have abilities, you will be valued. Yes, there are bias and prejudices in all walks of human life, But when things don't matter, there's a lot more room for those to impact things, like advancement or critical acclaim. Because it's so important, if you're in science and you bring the goods, you will be recognized and rewarded. Because you're going to learn so much this semester, because it's going to require a lot of work, I like to end this lecture with 3 slides showing you the payoff. I've picked 3 slides that we will encounter later in the course. I will give the lecture component of them that I will give later on in the course. You will not understand, I won't say not a word, but you will understand very little of what I'm about to say. But at the end of the semester, you'll understand every word. I do this because I think you'll see it'll be worth doing all the work that you're about to perform. Okay, in this slide, we have a schematic that shows how the cell captures energy from food and converts it to a useful form. Digestive foods from sugars and fats in the form of pyruvate and fatty acids will be transported from the cytosol into the inner mitochondrial matrix, in which case they'll be converted to acetyl-CoA. Acetyl-CoA will enter the citric acid cycle, in which the organic molecules will be oxidized in successive steps, producing carbon dioxide and capturing high energy electrons on the electron carrier NADH. NADH will donate the high energy electrons to the electron transport chain present in the inner mitochondrial membrane. Successive electron transfers will pump protons from the mitochondrial matrix into the inner membrane space. This proton gradient will then be used to generate ATP from ADP using the inherent energy contained in the electrochemical gradient. These coupled vectorial processes of electron transport, proton pumping, and ATP synthesis constitute oxidative phosphorylation, an oxygen-requiring process where oxygen is the terminal electron acceptor producing water. This is the way cells are powered. Stem cells offer the hope of greatly improving gene therapy. One of the difficulties that has occurred with current approaches for gene therapy is in order to repair a faulty gene, a therapeutic gene is incorporated into a plasmid, placed inside a virus, and then the virus is introduced into the target organism in an attempt to infect a large proportion of cells to replace the faulty gene with the repair gene. Now, in addition to immune responses from the virus, it's hard to infect enough of a tissue to get a widespread and sustained response. With stem cells, we are able to isolate them from an individual, place the therapeutic gene back into the virus, infect the stem cells, introducing the genetic material, grow the stem cells up, and then introducing the stem cells into the tissue, and they can then naturally repopulate the organ, including the corrected copy of the gene throughout the tissue. In this slide, we talk about 6 hallmarks of cancer cells that are different compared to normal cells as defined by Hanahan and Weinberg. These 6 capabilities that cancer cells acquire lead to devastating diseases affecting anywhere between a third and a half of the population. First and foremost, cancer is inappropriate growth, and normal cells are given growth signals in order to divide. Cancer cells have learned to become self-sufficient, growing when they're not supposed to, growing in the absence of growth signals. There are many brakes on the growth of normal cells, including crowding. Cancer cells have become insensitive to anti-growth signals, and they can grow uncontrollably, displacing healthy tissue. When normal cells are damaged to the extent that cancer cells are, they will commit a form of cellular suicide called apoptosis. Cancer cells have learned to evade the apoptotic signals and continue to grow even though they are too damaged to grow healthily. Normal cells only divide a certain number of times, as defined by the Hayflick limit. Cancer cells have learned to become immortal, grow, and divide an essentially infinite number of times, primarily by developing mechanisms for telomere elongation that enable it to grow long after normal cells would have stopped. Cancer cells have learned to invade normal tissue, to break through extracellular matrix, to metastasize in the bloodstream to other locations. Normal tissues do not invade other tissues and are anchorage dependent, meaning they do not survive when detached from the extracellular matrix or stroma and do not survive in the bloodstream. Finally, when cancer grows and creates new tissues, it needs a fresh blood supply. It has learned to control the growth of new blood vessels to support its growth, giving it the nutrients and metabolic requirements required for growth. These 6 capabilities that cancer cells require multiple mutations to many genes. And perhaps cancer has been so hard to treat because quite often therapies are only directed towards one of these capabilities. Okay, that's all I'd like to say for this video. I hope I have gotten you excited for what we're going to learn this semester, and I hope you are able to take advantage of this opportunity to learn this incredibly interesting subject matter at this critical time in human history.